Hello and welcome to Sam Biology class. Friends, today we shall discuss about the linkage map. A linkage map is the condensed graphic representation of the relative distance between the genes and their order of arrangements within the genome. By this technique, only the linked genes that occur in two or more heritable forms called alleles can be mapped. Linkage map is based on the analysis of recombination frequency, hence it is also known as recombination map. Before discussing further on gene mapping, here are some important points. Number 1. In a chromosome, genes are arranged in a linear manner. In a chromosome, genes are arranged in a linear manner. And number 2, all the genes that are located in the same chromosome are transmitted together in order to maintain the parental combinations of genes. This tendency of the genes to be transmitted together is known as linkage. And the genes that are located on the same chromosome are known as linked genes. The number 3, occasionally linked genes do separate by a process called as crossing over, by which reciprocal segments are exchanged between the non-sister chromatids of homologous chromosomes. And number 4, the percentage of crossing over between genes is directly proportional to the distance between them. So the genes which are widely separated have a greater chance of being exchanged by crossing over. But the genes which are closely located have a lesser chance of getting separated. Now let us draw a small linkage map showing the relative positions of three different genes and the distance that separate them. The genes are A, B and C. In a linkage map, the distance between the genes is expressed as arbitrarily defined unit known as map unit. Or MU. One map unit is equal to 1% crossing over. 1% crossing over, and 1% crossing over is equal to 1 centimorgan. So 10 centimorgans will be equal to 1 decimorgan. In a linkage map, the distance between the genes are expressed in terms of centimorgans. So in this map, the distance between A and B is 2 centimorgans and that between B and C is 1 centimorgan. A 3-point test cross is a more efficient technique of gene mapping than a 2-point test cross. To examine gene mapping with a 3-point test cross, let us consider 3 recessive mutations in the fruit fly Drosophila melanogaster. These mutations are related to eye color, body color and the bristles. In Drosophila, bristles are the mechanoreceptors that are components of the peripheral nervous system. These 3 recessive mutations are like this. Number 1, scarlet eyes. Scarlet eyes are recessive to wild type red eyes. Wild type red eyes. And number 2, ebony body color. Ebony body color is recessive to wild type grey body color. And number 3, presence of spineless bristles. Presence of spineless bristles is recessive to wild type normal bristles. Wild type normal bristles. To map these three genes, we have to determine their order of arrangements and the genetic distance between them. For this, we must set up a 3-point test cross between a Drosophila which is heterozygous at 3 loci and another Drosophila which is homozygous for recessive alleles at all 3 loci. In many organisms, it makes no difference whether the heterozygous parent is male or female, provided that the genes are autosomal. But in Drosophila, no crossing over takes place in male. Because crossing over in the heterozygous parent is essential for determining recombination frequencies, in our example, the heterozygous parent must be the female. Hence, this test cross is performed between a female Drosophila which is heterozygous at all 3 loci and a male Drosophila which is homozygous for recessive alleles at all 3 loci. To map these 3 genes, we have to know where and how often crossing over takes place. Because in the homozygous recessive parents, at 3 loci all the alleles are recessive, so crossing over will have no effect on the type of gametes produced with or without crossing over. But because in the heterozygous parent at all 3 loci both dominant and recessive alleles are present, so crossing over will be detected. As a result, the information that we require for mapping comes entirely from the gametes produced by the female heterozygous parent. And in this case, because all the alleles in the gametes produced by the homozygous parent is recessive, so the alleles that are present on the chromosome of the gametes produced by the heterozygous parent will be expressed in the progeny. This figure shows a 3-point test cross between a female Drosophila which is heterozygous at 3 loci and a male Drosophila homozygous for recessive alleles at all 3 loci. This test cross can produce 8 phenotypic classes, some of which are non-recombinants with parental combinations of genes, and remaining are recombinants with non-parental combinations. These recombinant progenies are produced as a result of crossing over. In all these, we have placed the gene for body color at the center. This is arbitrary because we don't know the actual order of the genes. After test cross, the next step is to identify the order of the genes on the chromosome. For this, let us identify non-recombinant progenies and double crossover progenies. Non-recombinant progenies can be identified based on the fact that in a cross they are the most numerous, even if crossing over takes place in every meiosis. In this example, these 2 phenotypic classes represents non-recombinant progenies. They are the most numerous. Then identify double crossover progenies. In a cross, the probability of double crossover is always less than that of single crossover. As a result, double crossover progenies are the least numerous. So here, 7th and 8th phenotypic classes represent double crossover progenies. They are the least numerous. In this example, we are mapping 3 different genes. These 3 genes can be arranged in 3 possible order. Now let us draw the chromosomes of the heterozygous parent showing three possible orders of genes. So these are the three possible order of arrangements of genes. In one, the gene for eye color is at the center. In second, the gene for body color is at the center, or in third, the gene for Bristol would be at the center. Now draw the homologous chromosome. The homologous chromosome have recessive alleles. To understand gene mapping, we should have some basic idea about linkage and crossing over. The recessive alleles are like this. So these are the three possible arrangements of genes. Then perform double crossover and see that how the order of the alleles has been changed. Now let us draw the resulting chromosome after double crossover. So these are the three resulting chromosomes which are produced after double crossover. Now let's see if any one of the resulting chromosome with a change in arrangement of alleles is observed in the double crossover progeny. By comparing, it becomes clear that it is the third order of arrangement of genes is observed in double crossover progeny. So the gene for Bristol must be present at the center. Now we can write down the correct sequence of the gene. Out of three genes, the gene for Bristol is present at the center, and on either side one is the gene for eye color and the other gene is for body color. So this is the correct order of the gene. After that, let us compare non-recombinant progenies with double crossover progenies. If we will compare first phenotypic class with seventh phenotypic class, then it becomes clear that both have the wild type alleles for eye color and body color. Only the difference is in relation to the locus for Bristol. Non-recombinant phenotypic class have wild type alleles for normal Bristols, whereas this double crossover progenies have locus for spineless Bristol. We'll get the same result if we'll compare the second phenotypic class of non-recombinant progeny with the eighth phenotypic class of double crossover. So by analyzing this arrangement of genes, we can clearly say that out of three different genes, it is the gene for Bristol which must be present at the center. So this is the correct order of the genes. After determining the correct gene order, let us rewrite the phenotypes. It is already done. The next step in gene mapping is to identify the location where crossing over has taken place. Out of 8 phenotypic classes, these two classes have already been identified as non-recombinants and these two as double crossover. Non-recombinant progenies are most numerous and double crossover progenies are least numerous. Remaining 4 phenotypic classes have been produced as a result of single crossover. So when crossing over takes place between ST and SS locus, these two phenotypic classes are produced, and when crossing over occur between SS and E locus, these two phenotypic classes are produced. The red slash indicate the location of crossing over. The last step in gene mapping is the calculation of map distance between the genes. Map distance is calculated on the basis of recombination frequencies. So let us calculate the recombination frequency for ST and SS locus. So while we are calculating the recombination frequency, we must include both single crossover as well as double crossover. So for that we have to add 50 plus 52 plus 5 plus 3. So here we have added 50 plus 52 plus 5 plus 3, then divide by total number of progenies, which is 755, then multiply by 100%, and the result is 14.6%. This is the recombination frequency between these two locus. So now the distance between ST and SS locus is equal to 14.6 map unit. Similarly, we can also calculate the recombination frequency for SS and E locus. So the frequency is 43 plus 41 plus 5 plus 3, then divide by total number of progenies and multiply by 100%. The result is 12.2%. This is the recombination frequency. So the distance between SS and E locus is 12.2 map unit. Now we can prepare the complete genetic map. This is the complete genetic map in which the locus for Bristol is at the center, and this is the locus for eye color, and that is the locus for body color. The distance between ST and SS locus is 14.6 map unit, and the distance between SS and E locus is 12.2 map unit. 12.2 map unit. After this, the genetic distance between the ST locus and E locus is calculated by adding these two values. So the distance between ST and E locus is 14.6 plus 12.2, that is equal to 26.8 map unit. This is the complete linkage map. Friends, for more update, please subscribe my YouTube channel, Sam Biology Class.